we will start with Margot Paulino's presentation. I would like first to introduce Margot. Margot is PhD in chemistry, and she studies the design of drugs, their mechanism of action, and the biomolecules involved. She has a passion for research in a multidisciplinary, challenging, and complex scientific area. Margot's happiest moment of the day include training successful professionals and scientists and learning at every step with her 3 wonderful children and 2 grandchildren. So, Margot, if you can share your presentation, we are very happy to have you here today.
Okay, thank you very much. Thank you very much, all of you. And I'll present a part of our research line related with the articulation of many tools to the innovative design of bioactive compounds. First of all, I thank you very much for organizer for this kind invitation. My university to support this research, and a lot of people, including some of our postgraduate students, as Andrés, Jorge, Pablo Garcia, colleagues, Pablo Denis, people from the Instituto Pasteur Montevideo, from the Universidad de los Andes Bogotá, Pontificia Universidad Católica de Chile, Universidad Nacional de Córdoba, people from Faculty of Science in our university, and two ex-students like Brenda Vera and Mayan Navarro. Mainly Brenda has a very good time with us and in her master in bioinformatics, and Fabio Polticelli and Andrea Cavalli as a consultant. Okay, now I'm concentrate my talk in the parasitic disease that are a major obstacle in health and economic development, with the high rates of mortality and morbidity, therapies and satisfactory treatments, failures, and severe side effects. So we couldn't solve this problem, and we have the necessity to develop or to improve our candidates for drugs. In this particular case, trypanosomatids like Trypanosoma cruzi, Leishmania, Trypanosoma brucei has many stages with many metabolic behaviors, and are, in the case of Chagas disease, it's transmitted by the kissing bug, is a vector, and as I show a picture of 2015, is an endemic disease in all around South America and Central South America, and is present in all North America, Europe, and other continents. We support a main hypothesis that is that specific imbalance of the normal redox balance or oxidative stress is one of the preferred ways to fight against trypanosomatids. Trypanosomatids had a very complex system to the redox balance, and have some differential enzymes that are targeted by a lot of people. And in this sense, quinones is a scaffold that has demonstrated to be apt to the development of new drugs for this disease or other trypanosomiasis, because its putative capacity to induce oxidative stress. So the hypothesis is that quinones could induce oxidative stress, or they could modulate the redox level of parasites, modulated oxidative stress enzymes in the parasites and in the human. So we need to compare all the time the capacity of quinones to produce itself the stress, oxidative stress, and the capacity to inhibit targets in the parasite and in the human. Quinones are an example and they could form as a first step a semiquinone capturing one electron, and in a second step a hydroquinone in the propagation step, and then they finished with this kind of reactions, and in the middle oxygen forms the oxidative stress oxygen species. Then our objective is to revise the oxidant and non-target capacity of quinones, and to revise the modulation of the reactivity of redox metabolism enzymes in the parasite and in humans. These are three of putative targets: trypanoside reductase, GPDH, G6PDH, and lipoamide dehydrogenase, selected to do at this moment biochemistry. But we will see that there are a lot of available and putative targets, making quinones as a lot of other scaffolds very reactive in different metabolisms, so multi-targeted actions. And some promiscuity in the sense in which we cannot to modulate just one enzyme and not consider others enzymes and metabolism. We have a very good support in the wet lab, that is a group making synthesis in Chile, making epimastigote growth control in Uruguay, tripomastigote growth in Institut Pasteur, as well as the development of redox probes. There are assays over entire tripomastigotes, and the biochemistry, as I told before, of GCPDH or other enzymes. Respect to the TR biochemistry, TR is one of our old targets. We made yet a biochemistry in Germany with Karina Vázquez and Professor Kraut-Siegel. In the in silico lab, we developed with the time, and more recently too, a lot of tools that are ligand-based, so quinones-based, reverse screening, artificial intelligence, and structure-based drug design. LBDD consists in modeling, alignment, and design, for example, of pharmacophore, and to do quantum chemistry to define or to measure electronic global descriptor. To detect targets, we developed two kinds of methodologies: reverse screening, and now we are using artificial intelligence to the detection of targets and new design of bioactives by deep learning and reinforcement learning. And in the so-called structure-based drug design, that is based on the target, we are modeling the complete sequence of many, for example, in the case of G6PDH, in the PDB is the monomer, we designed the tetramer, we complete the tetramer. We are doing docking by many ways, and molecular dynamics, mainly with NAMD in the CUDA version, and finally measurements of binding affinity by the endpoint method. Some results in LBDD. Now we are working with a 62 sample with a very variable set of scaffolds: naphthoquinones, pyridine-quinones, tetrahydrofuranquinones, oxazole-quinones, furanquinones, and thiazole-quinones, with different kind of wet lab. It's not the only sample, but I concentrate the description of our results in that 62 sample. For example, save those main scaffolds, the series has variation in a lateral substitution that in some cases are two lateral right and left substitutions with a lot of proposals, so making more capacity to keep hydrogens, some diversity in charges. Some of them, here is not an example, but with nitro groups with a very strong electrostatic charge. And In the beginning of this research, we found a very good inhibitor of T-cruci with an IC50 of 20 nanomolar, yes, and with a very good selectivity index, because we checked the macrophages reactivity and was very bad. So we pointed to that kind of compounds. We have a diversity of similar compounds with, as you could see, different results in the selectivity. As a data, nifurtimox, one of the marketed drugs with very severe side effects, has a selectivity indexes of 45. With respect to our research in the electronics description of these compounds, we evaluated the 3 stages, the neutral, semiquinone, and hydroquinone stage. And now I'll show to you the semiquinone, because our hypothesis is that those semiquinones are the reactive species. We evaluate global indexes by DFT with this kind of functional. And that was the sample that I showed last year, but it was amplified by that I described below. We have naftoquinone and all the scaffold put here, and you could see the big diversity of IC50, some of them very good, some of them very bad. So if we see those under 10 micromolar, we have this kind of distribution. It is apparent that naftoquinones and pyridinquinones could be the best scaffold. So when we cross data and we obtain the SUMO, so the high half occupied orbital, frontier orbital in the semiquinone stage, named SUMO, and we normalize it, and we related with IC50, we could appreciate that in this case, naftoquinones and some pyridinquinones could be related. Looking for some correlation, we found a 43% of correlation, or R-squared correlation of 43.43. That is not so good, but is enough to think that these semiquinones could have a part of their reactivity in the parasite caused by the formation of that semiquinone. The molecular orbital, we selected the best and the worst. You could see that this is a very new result. Actually, Andrés Camilo, that is making his PhD thesis, give this result yesterday. We could see that the SUMO orbital is distributed over the naftoquinone in the best naftoquinone, and is spread over one of the substituents. So we think that is more energetic, so is more stable and more spread, because this is the less energetic label, so more reactive. Okay, referring pharmacophore, I think all of you know what is a pharmacophore. It's a graphical description of some very important properties related with the pharmacology of molecules. The naftoquinone has 2 acceptor regions in the quinone, 1 more, and here a lot of things. But when it appeared in the heterocycle as third acceptor, we observed that the activity was worse. And when in the case of oxazolquinone and thiazoquinone, they was 4 acceptor of hydrogen bonds, the result was very bad. So there is a conclusion there. Referring the structure-based drug design, first of all, we put a hypothesis, they have a target, a TR, etc. But we wanted to be very rational in the selection of targets. So we developed the so-named target fishing methodology by shaft that looks for similar ligands co-crystallized in the PDB, and by a FarmMapper that is looking for similar pharmacophores between the pharmacophore of the molecule and the site in the crystallized protein in the PDB that in this case could have or not a co-crystallized ligand. So this is our preferred method those days because we have more data, because we have a FarmMapper has a very good database of pH4 sites. But then we fish hundreds of proteins and we filter it. How we filter it? Looking for those targets in a property and specific trypanosoma database, trypanosomatic database, that is a TRITIP database, and in a repository of scientific journals, looking for the names, the key names that could orient us the search to the trypanosomatids. As a result, we detected at least 11 emerging targets. One of them, the trypanosine reductase. So the hypothesis that the TR is a target is sustained too by this method, but they appear other targets and other metabolism. So multi-targeting and a promiscuity in the action that could be taken as an advantage, because if the quinone impact over many sites could be more effective. But the problem for the designers is that we need to know that and the structure related with that multiple activity. So in this part, we started from a second point of view. So if our redox is our preferred metabolism, or if our main metabolism is the redox one, then go to KEGG database and look for specific proteins of Trypanosoma cruzi inside those maps. And this is a part of the PhD work of Andres, and he found, looking for redox enzymes, many metabolism, filtering by TRITIP database, and others, amplifying for trypanosomas and leishmania, because both of them are trypanosomatids, and selected oxidoreductases by the EC number. We found 32 proteins. In the meantime, we verified a second time that the metabolism of nucleotides appears too, biosynthesis of terpenes and steroids. So there is a metabolism alternative that could be exploited in the search. But now we continue with the main redox metabolism. So we are founded those 32 proteins. This is the list. You could see here the TR, here the dihydroorotate dehydrogenase that appeared too in the shaft previous search of Brenda in her thesis, etc., etc. For example, that one triparedoxine peroxidase that is related with trypanosine reductase 2. After that, we build the complexes between those targets and those ligands, and we apply a molecular dynamics generic protocol with all of classical steps: minimization, annealing, equilibration, sampling of different sizes, and repeating sampling too. Always using a time step of 2 femtoseconds and sampling each 2 picoseconds or more through all trajectory, and then analyzing as usual. And finally, when we detected the equilibrium and we are sure about the complex structure, we make a binding affinity evaluation by more precise endpoint methods. One of the results with trypanosine reductase, this is the protein involved in or linked to the triparedoxine and triparedoxine peroxidase metabolism with several, in each case, several substrates, but trypanosine is central in that. This is trypanosine, is glutathione, linked to spermidine amine in the oxidized and reduced form, and making a massive docking over all the structure of TR, and making biochemistry, we found some of the naphthoquinones with a preferred inhibition of TR than the mal counterpart, that is glutathione reductase. This is a partially successful result because we couldn't improve the selectivity of compounds over the TR, but we are in the way. We found a very new site for the anchoring of these quinones that interfere with the anchoring of natural substrate. This is a trypanosine. This is our site founded for the naphthoquinones that is justifying a competitive kinetic found in the biochemistry. So they interfere with the binding of the substrate by there is enough room in the enzyme to bound both substrates. This is in agreement with the biochemistry made by Karina in Germany. So then we are founding naphthoquinones that are acting by a multi-targeting, and in the future could be a good objective to reposition it or reevaluate those quinones for another uses in for other diseases. And there is a very interesting talk after mine about this subject. So conclusions. It seems that just 2 acceptors in the quinone ring are recommended, and in the case of the semiquinonic naphthoquinone rings, they only clearly related with IC50, respecting the SPDD-TR, is partially inhibited with some specificity, multi-targeting mechanism, and reverse screening amplified our vision about our kind of targets involving our metabolism, not only glycolysis, but lipid or vitamin and nucleotides metabolism. Okay. All in all, naphthoquinones remains a good one, or the good one, to continue the design of good trypanosomicides with a putative capacity to produce a redox imbalance in the parasite. Finally, artificial intelligence. Why we want to do that? Because I have a very, very, it's very good student as all that I have, but Pablo is special because he came from the informatics field, is an engineering, and he's making the master in bioinformatics. So he migrated from the informatics to the chemistry, a big effort. And now we are finishing this thesis with some results. Challenges to acquire data on interaction of molecules with bioactivity in proteins, to represent the information in such a way that the neural network can use to learn, so to teach the neural network with that information, to generate new ligands, to transfer knowledge in a way in which we could improve the capacity of the network, and to optimize new ligands until they can be validated by, finally, traditional bioinformatic methods. we are using this kind of tools and some results. The annotated PDB bind, so we are using a PDB bind that we are annotated. That database has 80,000 data, usable biomolecules, ligand pairs. We are using FPocket to define the anchoring sites in a generic way, in a way to that the network learns the protein ligand interaction, how they are. A network convolution model is making possible to encode both the ligand and the anchor site from the molecular structure. Second, we are using a variational autoencoder to generate various ligands for each target site. And we use transfer learning to initialize a new network which works only with a defined target. Finally, a reinforcement learning process to feed back to the previous network and generate more information, and to filtering by docking Lipinski rules, ADMET, and some very empirical descriptors like synthesability, originality, among others. Okay, and now I'm finishing with that line. I talk about two new challenges that we have. One with the Universidad Nacional de Córdoba that are researching with sesquiterpenic lactones. That molecules are not redox active, but they could be impacted by binding to the enzymes in the redox metabolism. No, this is the proposal of these people. And then the challenge was to modelize these sesquiterpene lactones came from natural products.
Sorry, Margot, we are on time.
Okay, so I just mentioned the last challenge. I keep that, and I want to mention people working in the trypanosine synthetase. So sorry. I voy rápido. Acá, this is the team working in trypanosine synthetase. We model it, and we are making research with, in this case, different products that forms covalent bond. This is the very special thing that I would mention, that we need to develop a covalent docking to solve this problem. So thank you very much, Wendy, and I'm here to question or to talk after the conference. Thank you very much.
Thanks a lot, Margot, and sorry for interrupting you, but we have another speakers, so we have time for one question. Please leave in question and answer, or raise your hand. So I don't see any question. I have two questions, Margot.
Okay.
So the first is, how do you measure the selectivity index of the compounds?
Okay, it's a ratio between the IC50 in macrophages or another kind of model to measure the cytotoxicity, and the IC50 in the trypanosomatids.
Okay. Okay, and I missed the name of the program that you use to identify pharmacophore ligand and protein simultaneously.
Yes, this is the FarmMapper. FarmMapper is actually it's not a program. It's a site that could be used. Could not be used on site. Could be used in the site.
Okay, perfect.
FarmMapper.
So, and we have one question. Okay, there is not There is not a question, it was all, sorry. So I think that, let me check the question and answer. Okay,
Okay.
what tools do you recommend for docking for compounds on Trypanosoma cruzi targets?
Okay, thank you, Mercedes, and all of you for your comments. We could talk after Mercedes because it depends on the complex. We are using MOE program that is a small level and it's not so high level, listen set and all of that. And when we are, the result that I showed to you was obtained with MOE, but now more recently we are using Autodoc. It's more guarantee program, but I recommend MOE if you could buy the license.
Okay, thanks a lot, Margot.